Yeah, warm good afternoon to everyone here, the speaker, and all the participants in the today's talk. I am Ravi Kiran, and I am a member of the Science and Technology Council. And this year, Anvesha is proud to be sponsored by Sabari Scientific Supplies, Apparatus, and Chemicals. We have organized this Nobel talk in collaboration with with Proteus, the Biology Club of IISER Thiruvananthapuram, and I would like to welcome Charu to introduce the speaker to you all and begin the talk. So I'd like Charu to request to continue with the introduction. Thank you.
Thank you, Ravi Kiran. Hello, everyone. This is Charulekha on behalf of Proteus, the Biology Club of IISER. First of all, I would like to welcome you all to today's session. We are going to have the second talk on the Nobel Prize Talk series organized by Anvesha, the Science Club of IISER in collaboration with Proteus. This year, the Nobel Prize in Physiology or Medicine was awarded jointly to Harvey J. Alter, Michael Houghton, and Charles M. Rice for the discovery of hepatitis C virus. Today, our speaker is Dr. V. Stalin Raj from the School of Biology, IISER. He holds a PhD in biotechnology, pathology, and microbiology in aquaculture from the University of Madras, Central Institute of Brackish Water, Aquaculture's ICAIR, Chennai. He was a postdoctoral fellow at University of Liege, Belgium. He worked as a scientist at Erasmus Medical Center, Rotterdam, Netherlands, from 2010 to 2017, and then he joined IISER in 2017. Today, we will revisit the Nobel Prize-winning discovery with Dr. Stalin Raj, who will be giving us an expert exposition on the research and the significance of this year's Nobel Prize. I'd like to now welcome Dr. Stalin and hand over the mic to him.
It's okay. Thank you, Charu. So it was a great introduction about me, and today I'm going to talk about the Nobel Prize for the Physiology or Medicine 2020. So for the discovery of hepatitis C virus. Okay, actually this is very simple. You may think, for the discovery of a small virus, they're giving a Nobel Prize. And I will briefly explain to you the details, what are the struggles they face, and how and what is the purpose they got the Nobel Prize. This year there are 3 people got the Nobel Prize. One is Professor Alter and Houston and Professor Rice for the discovery of hepatitis C virus. You all know, no? This is our liver, no? This is a liver. This is one of the second largest organ in our body. It has more than 500 different functions, and it play a major role in the fighting against infectious diseases. And also they produce proteins that require for the protein and also cholesterols. And also they excrete waste via bile, and also they produce protein that help for the digestion, and also to maintain the glucose level in our body. And in addition to this, the liver also play a role in blood clotting. So this, all of them is actually more, but I put them, just a snapshot I took it from the internet. So it's so much of functions will be done at our liver. Is only done by there is a cell called hepatocytes. Hepato means liver, cytes means cells. That means liver cells. These liver cells is responsible for making this all type of functions. Okay, so if something happen in the liver, what will happen? For example, if your liver is become non-functional due to the beverages or drinking beverages or toxin, some medications, or due to some medical condition. For example, medication means if you take paracetamol, too much of paracetamol, that also can cause hepatitis. Okay, they can damage your livers. most importantly, some of the viruses that can damage the liver cells, your livers. Okay, so there are two types if you have a hepatitis. There is one is in the acute stage, and the other one is a chronic case. In the acute or chronic both case, is most of the cases, if you have a hepatitis, you mostly you don't have any symptoms in the early stages. That means asymptomatic. Later, become symptoms. In the case of acute, later you have a symptom. Then when the time goes, you will have like a liver failure. At the end, you have a death. Okay, you have a symptoms. What are the symptoms? You can see nausea, or vomiting, abdominal pain, loss of appetite. You have fever, diarrhea, and dark, clay-colored stools, dark urine, by example, in yellow in color because of bile. So that, or jaundice, actually you have skin or your eyes, you can see in a yellow color because of the sediment of bile on the skin. So this is, in the case of, this is actually happen in a short frame, in the acute case around within 6 months it will happen. But in the case of chronic, is a chronic hepatitis is not a short term, it's a long term. It will be start from the normal case, then you will slowly within in 6 months it will be reached a chronic hepatitis. Then later in 20 to 25 years you will have a cirrhosis. Then at the 25 to 30 years you have a liver cancer. Okay, so ultimately, if you have anybody or somebody have a liver disease with a cirrhosis, or if they become a liver cancer, the end result is mostly to have a death. There is no therapeutics or vaccines available for this type of diseases. If you have a severe cases, if somebody have a cirrhosis, you have accumulation of water in the belly, for example, edema. That means, for example, I told you one of the major function of our liver is to produce production of albumin. The role of albumin is maintain the osmotic balance, water balance in our circulatory system. If there is a less amount or reduction in albumin, actually the water molecule that can permeable or permeable from the vasculars or the circulatory system to the tissues actually. In the case of here, what will happen if there is if somebody have a cirrhosis, at the end of the day there is a lack of albumin productions, then the water molecules then they're secreted into in your belly. So at this stage you cannot do anything. The only way you can take it out, usually the doctors put a hole on your belly and remove the water from there. But the treatment, there is no treatment available for them. At the end, the result will be like this. The complication is like this. At the end, there is the only way you can cure is you can do a liver transplantation, but almost impossible to have a liver transplantation because whoever have a cirrhosis, they can recombine back on cirrhosis. So it's very difficult to treat. End result is actually death only. Okay. So if you think about cirrhosis, there is is a end stage is called a cirrhosis. You can see, there is no therapeutic or available to date, and the only option, as I said, is liver transplantations. But now currently there are people working on stem cell therapy, but there is is in the early stage of actually what they do, that they put the stem cells implant into human liver tissues, or not a human, actually they're testing in animals. Then later they can they could able to recover the cirrhosis. They become normal. So this also will take quite a long time to get back to this type of therapeutics. So as I mentioned, the cause of the hepatitis is normally toxins or other diseases also play a role. One of the common cause for the hepatitis is viruses. There are 5 different types of viruses that can cause viral hepatitis. Hepatitis 1 is hepatitis A virus, hepatitis B, hepatitis C virus, hepatitis D, and hepatitis E. These See, one of the important point here, these all 5 different viruses, these are not belonging to the same family. All 5 are from different family. They have different type of genetic material, but they do the common, disease. They cause the common disease is hepatitis. Okay, this is a very important point I want to mention here, because the most common one is hepatitis A, hepatitis B, and hepatitis C virus. So hepatitis A, I think you all know, when you are in a young age or small age, you might have got a hepatitis, jaundice you often come, is caused by hepatitis A virus. It's mostly they transmitted through oral route. It's actually when you drink, high contaminated water or food, there is actually basically less hygiene, or if you use drug, if you use syringe or needles, or then that also you can transmit. And also homosexual people can mostly get this kind of hepatitis. But this is where it is an acute infection, and this is curable. There is a vaccine available. It is preventable. Once you have a vaccines, or if you get one-time infection, you have a lifelong immunity against this virus. The second virus is the most important virus. Is a hepatitis B virus. This is a sexually transmitted virus, or they transmitted through blood product or blood product, which is 100 times more than the HIV. This virus, they also transmit either blood, is a bloodborne, either sexual transmission or injection, reuse of needles, tattooing, or mother to child transmission also there, and also this become chronic infections. But for this, at this moment, for this virus, the vaccine is available, but you can also prevent hepatitis B virus. So third virus I want to talk to you is hepatitis C virus. Actually similar to the hepatitis C virus. There's a transmission route and everything similar to hepatitis B virus. But mostly this virus transmitted actually between drug users. Actually, is primarily on who use drugs. Those people, drug users, they use the same drug or same type of equipment, and they use for the drugs. Okay, so someone else use one needles, and then they gave it to the another. Multiple people use the same needles or equipments. They will get the hepatitis C virus. But for this virus, there is no preventive, there is no vaccines or therapeutic available, for this virus. So if you look at the why it is very important, more than 325 million people have been infected with this the hepatitis virus. Around 250 million people are had a hepatitis B virus infection. Around 70 million people worldwide had a hepatitis C virus. So overall, if you think about it, around 1.4 million people die from viral hepatitis, including cirrhosis and liver cancer. Compared to COVID-19, think about this. We are not worried about other viruses. We are only thinking about COVID. Actually, there are many other viral diseases and also bacterial diseases are much more pathogenic than the COVID as well. Okay, everybody has to think that as well. There is a tuberculosis or hepatitis, the hepatitis C, HIV, hepatitis B, hepatitis C. These are, they cause, high mortality. In this case here, now among the 1.4 million, around HBV and HV is responsible around 90% of the fatalities. Around 10% is only, for other caused by other viruses. Okay. So if you look at the graph I showed, the acute, hepatitis, viral hepatitis in United States, estimated around hepatitis A virus 93,000, hepatitis B 78,000, hep C is 25,000, but hep D virus is relatively less. But one thing you have to do, hepatitis E virus is, as I mentioned to you, acute, never reach even chronic infection or chronic liver failure, whereas hepatitis B and hepatitis C virus, they undergo for chronic infection and also leads to have a death as well, high mortality rate. But so that if you look at the transmission route of the viruses, hepatitis A, I told you, as I mentioned, A and E, a virus is transmitted through contaminated water or fecal route, and B, C, D is transmitted through the blood-borne, through blood or contaminated blood. And the prevention is, for example, hepatitis. You can see hepatitis A, you have a vaccine, or if you're exposed to the hepatitis A virus, you can prevent. You have immunity against that virus. Hepatitis B virus already have a vaccine against this virus. And the hepatitis, but only thing is hepatitis E, there is no vaccine, but you can control by, the preventing through, to avoiding contaminated water. But, and also hepatitis D also have a, if you have exposed to hepatitis D, where you have immunization. Whereas hepatitis C virus is a high risk, but there is no vaccine. So I will tell you in the later why there is no vaccine, because I told you we have vaccine for all viruses or therapeutic for all viruses. Why not for hepatitis C virus? Okay, so this is why it is important. Is 2.7 million people, even United States alone, have a chronic infection, and more than 10,000 people died because of hepatitis C virus infection. Okay, so hepatitis C, what is hepatitis C is, this virus is belonging to the family of flavivirus. Is similar to the dengue virus, no? Dengue, Zika, West Nile, yellow fever virus. If you look at the phylogeny, this is a whole flavivirus family. But if you look at the hepatitis C is here in the order of Hepativirus, and the family hepatitis C virus. Okay, so this virus is similar to other flaviviruses. So we have a structurally similar to other viruses. It has a single standard positive sense RNA genome surrounded by a capsid protein, and also on the top you can see a envelope protein. And again on the top, nicely decorated with a protein called envelope protein called E protein, envelope glycoprotein E1 or E2. So basically, the structure-wise, almost all viruses have a similar kind of architecture. Only differences in the term what you use here, or here is in the envelope protein we say. Here we say E1 or E2 is envelope protein, envelope glycoprotein. In the case of HIV, we say glycoprotein 120, or in the case of if you want coronavirus, we say spike protein. Okay, the genome wise, it's a large genome, around 9.6 kilobase per genome. I told you earlier also, some other talk I told you, this is an average size in the flaviviruses. The coronaviruses have the largest genome, around 30 kilobase per size. Otherwise, all other viruses have a lesser genome size. This is around 9.6 kilobase per size. It is code for, it is code for a single polyprotein from, is a code for a single polyprotein. Once is translated as a single protein. It's a long polyprotein. Once we produce a polyprotein, there's a viral protein itself. They have a proteases. That protein cleave this protein into the structural and non-structural proteins. Okay, there are 10 different types of structural and non-structural proteins. They have a cleaved, and once it cleaved, they do their job. Okay, so if you look at the life cycle of viruses, as I earlier also I discussed about coronaviruses, actually similar to other viruses. The hepatitis C virus also enter into the cell through the receptor-mediated endocytosis. One of the complexities here is, the hepatitis C virus uses multiple receptors. It's not only a single receptor. They have different types of receptor, and also vary between cell type as well. So once it binds on the cell surface, they enter into the cells through the clathrin-mediated endocytosis. Actually, the clathrin adapters bind it and they make a vesicle, then take into the cells, inside the cells. Then once it goes in, then the genetic material is released in the cytoplasm. Since it has positive sense RNA genome, positive sense means it's actually exactly like a messenger RNA only. Okay, so because the positives have a ATG and lead a sequence, ATG and stop, and also polyadenylation cycle, they can directly can bind it to the ribosome and can produce proteins. Okay, that's the advantage for the positive sense RNA virus. That's the reason the positive sense RNA viruses replicate fast as well. Okay, because directly they can code for a protein. Meantime, then also they can make with the reverse transcriptase, and also they can make their own copy. Then with the protein, and then later they can with the replicate with the own genetic material, and also with the help of protein, they pack them and assembled in a vesicle, then released out the virus. This is a general overall, the life cycle of the hepatitis C virus. So one of the difficult part in hepatitis C virus, as I told you, most of the RNA viruses have a polymerases. Here also this virus have a polymerase. It's called, if you look at the genome, you might have NS5. Actually, the NS5 is the code for the polymerase. This polymerase, mostly all RNA viruses polymerase, it doesn't have a proofreading activity. Actually, during the synthesis, what they do is incorporate all the nucleotide sequences, resulted in a lot of heterogeneity in the genomes. You have huge diversity. You have a huge diversity in the genome. Later I will show you many examples. So because of this huge diversity, there are 7 different viruses. They are classified in 7 different genotypes have been classified. If, for example, one genotype 1, 2, 3, like that is 7. Within the one also several subgenotype also there. Okay, so that is make it more complex. In a single virus, you have a huge diversity within the genome level. Then making a vaccines or therapeutics is very difficult, and also this virus escaped from immune system very quickly. And also if you look at the distribution of the genotype, with a geographical distribution, so you see you have huge diversity between country wise. For example, in India, you see you have a genotype 3A and 1B. So actually there are 1A and 1B. These are the most common cause of cirrhosis, but other species other serotypes are not very problematic. The genotype 1B is the most dangerous one. Okay, so. And also there are the people they feel more the Hep C is more prevalent only in the drug users actually. So if you look at this as a global picture, where you see the prevalence of hepatitis C viruses in the drug users. So actually you see there's one place, I think you remember, or how many, I don't know, any of you are aware of this. This is Egypt actually. Egypt have a high proportion of the hepatitis C virus. Actually, what happened? They have a hepatitis. The vaccine campaign happened in Egypt. During the vaccines campaign, what they did, they used the needles, same needle for multiple people. Actually, contaminated needles used to multiple people. There are many people has got infected by the hepatitis C virus. Okay, so go back to the reason why these guys, Professor Alter and Houghton and Rice got the Nobel Prize. So early days, the 1960s and all. Like, we know the hepatitis, but there is no proper diagnostics for these viruses at that time. They classified the viruses based on the characteristics. Then hepatitis A and B was classified because they know is a hepatitis A already is a chronic infection. Is transmitted is is acute infection actually. They can identify very quickly. They're temporarily serious, but they will become cured quickly. In the case of hepatitis B virus, is this is transmitted through blood fluid or any other blood materials, or and also they have a long incubation period compared to the hepatitis A virus, and also this disease can go for a chronic. And this time even they don't know about hepatitis A and hepatitis B, because they classified, I mentioned here hepatitis A and hepatitis B. Actually they don't know about hepatitis A or hepatitis B, but based on the disease characteristic, they classified in two. Okay, so actually at the time, Harvey, Professor Alter, working at NIH, National Institutes of Health, he was working in the department of blood transfusion. They are doing a lot of transfusion for the people. He was always monitoring the people after transfusion, what is happening, what their health condition. He regularly monitoring it. One day, what happened during that time, the whoever people receive transfusion, actually previously before transfusion, these people have doesn't have any symptoms or any disease for the hepatitis. But after transfusion, many people have a disease kind of hepatitis. Okay, so at that time he also worked along with, there is a Professor Blumberg. Along with him, he was working, and they discovered the hepatitis B virus. And for that only, Professor Blumberg got the Nobel Prize for the discovery of hepatitis B virus. So later, actually, Alter still didn't do, because that time he was a young and he was working with them. Then he also identified the antigen is one of the major surface antigen of hepatitis B virus. He identified, actually he also presented in one of the symposium, liver, and you can see this is the first paper he explained about that one. Actually, using this protein, they can also develop antibodies. You can immunize them in animals, they can generate antibody against this one, with the help of this antigen, they can also develop vaccines, and later they develop their diagnostics for detection of the hepatitis B virus. Again, at the time, they only, this time they know, the diagnostics only for hepatitis B virus, and they also develop vaccines. With this help of this, the hepatitis B diagnosis, they could also screen people, those, the donors. Actually, they collect blood from the donors, then only they do the transfusion. Actually, they could able to screen people, those have their hepatitis B. They could able to eliminate those who have the hepatitis B positives. Okay, that was an important finding at the time. But actually, what happened that time, they could able to screen it. Actually, they could. But what happened at the end of the day, still even only around 50% only they could able to eliminate. But then still around 50% of the people still getting hepatitis. So one of the things what they thought it could be, the other type of is acute hepatitis. Now we call it hepatitis A. That's what they assumed that it could be a hepatitis A virus. And another group could be, during the screening, we will always get a false positive. Maybe we slipped from the initial screening. That's what we have getting a 50% of them positive. But that time, Alter and with his group, what they did another interesting experiment. And the experiment, they took a 22 patient. They also screened for the hepatitis antibody against this hepatitis, anti-hepatitis B antibody, using their diagnostic visit before undergo transfusions. So after transfusion, what happened? A lot of people got the hepatitis. When you screen for the seroconversion, for example, they detect for the antibody against this hepatitis B virus, they couldn't find the antibody against hepatitis B virus, but still they become positive. And also they didn't find anything for hepatitis A like antigen also, they didn't find it. So then that is the first report that Professor Alter told that that could be, this is something is, causing hepatitis, and that could be as a non-hepatitis A or, hepatitis B is a non-hepatitis A or B. Okay, that's what they named at the time. But as I said it in earlier, they could be able to screen initially. During, again, this still you were studying, and that during this study you could able to screen a lot of donors. You could able to exclude many patients who have a hepatitis B or A patients. Later, still around 80% of them are causing disease through unknown agent. Unknown agent means actually they, in the acute case they know that already, but in the chronic cases, they know this. It's actually, they know that it really is coming from the blood product, not through contaminated water or anything. And also they're causing chronic diseases, and this chronic diseases is much in adult, compared to much higher than in the hepatitis B virus. So then, Alton made a paper, and he proved that the agent is unknown, but he named them non-A, non-B hepatitis. How he did it actually? In his discovery, he collected blood samples. He screened for the hepatitis B or in advance. They all are on hepatitis B negative. Then they will undergo transfusion. Whoever developed, hepatitis symptoms, then he collected serum samples or plasma. Then he injected into the chimpanzee. What happened actually? The chimpanzee developed a similar kind of disease. Okay, similar kind of liver diseases, actually elevated level of ALT. If you have liver diseases, how you do? Usually when you go to the hospital, they do a liver function test. Actually, it's called the ALT. The first test they do that because alanine transpiration is enzymes. If there is a damage in your liver, this particular enzyme will be higher. So if it is elevated level of ALT, that means you have a damage in your liver. Okay, so that's what they did it. Actually, he found it in the the high level of ALT, and also when you after sacrification of the animal, then also if you take the histology of the animal, they could see the changes in the histological changes, tissue changes in the liver tissue. However, they could not find any serological evidence for hepatitis A virus or hepatitis B virus. So based on this, he's the one they proved there is there is an agent that causing disease is a hepatitis, but the causative agent is unknown. They named us maybe the non-A, non-B hepatitis. Okay, during that time, Houston was working at the time, working on that to discover the causative agent, because there are very few people working, very vigorously on hepatitis. Actually, this time he was working. Another experiment they did during this time, when they inject interferon in interferon. For example, they select 10 people. Then they inject interferon for up to 16 weeks continuously. Actually, the people who had a liver disease, they become normal. So one of the limitations what they found, when they stop it for sometimes or 4 weeks, then the virus reappear again. Okay, I will tell you the story why it is virus disappear. Then what are the possibilities can come back again. So one thing is that they will show that interferon therapy could lower the virus infection, and on the other hand, they will also get a clue for that could be a virus, because interferon therapy is interferon is mainly for the antiviral agent. So could be they got the clue. At that time, Professor Houghton is working on find out the causative agent for this virus. He tried all the possibilities. He tried, for example, he said that time, we tried everything. but everything is failed actually. Nothing. They couldn't find anything. So they tried to do all methods to prove the successful, for the other, what are the causative agent for this disease, but they couldn't. We could not find out any antigen or antibody against that for this particular agent. They could not find it, and also they could not able to grow the virus. And if you look at the electron microscopy, they also couldn't see anything on them. So that means at the time, because nowadays we have a lot of technology, that time you think about it, they tried all known techniques, but unfortunately they didn't find anything at the time. So what Hutton thought it now, so then he collaborated with another group, and he decided go for a different approach. You can go for a proteomic approach. So what he did, he actually, I told you, they inject them the Hep C in a chimpanzee. They collect the samples, plasma, and they pellet them. Once you pellet them, they extracted, nucleic acid. Actually, they clone them, everything on in the phage, bacteriophage is a phage, bacteriophage, and then put them in a bacteria, called the phage display library. Actually, they clone them. If you clone the genes, actually, on the bacteriophage, what will happen? The idea was they clone with the promoter sequence. Then whichever protein is expressed, always present on the surface of the bacteriophage. Okay. So then what they then they used the human, already those who are positive for non-Hep A, non-A, non-B hepatitis, the serum, they used to the fish out the positive clone. For example, if there is the whichever clone, they don't know whatever clone. They clone, they have around 2 million clones. That means around 20 lakhs clones. They put them on they try to find out, fish out the clone using that specific antibody against that particular the virus or particular other or unknown organisms. Interesting, luckily actually the one clone, they picked, they know the one of the antibody find it on the one of the clone. That was really positive for the hepatitis C virus. Actually, when they sequenced them, later they found the hepatitis is a genomes which is related to hepatitis C virus, is related to, it's a flavivirus. So later they named them hepatitis C virus. Okay, this is the way the first time he discovered the hepatitis C virus. Actually, this is at that time, we don't have any novel techniques. This is the they said this is a novel technique to identify the unknown organism. That is the reason he also got a Nobel Prize to imagine you have a 2 million clone to screen them. Think about how long it will take. So it is it's a huge effort to find out that the organism. So now we know the virus actually. Now we know this hepatitis C virus, we know the genome, but nobody knows, how to prove that this is a particular, this hepatitis C virus is the causative agent for that particular hepatitis. Very difficult, no? So usually, I don't know, many of you, my students, they know I was also taught about it, Koch postulate theory. If you have an infection, you have to infect them in an animal, then you have to, the animal also has to have a similar kind of symptoms and disease, what happening in the human. Then after, then you have to collect the samples from the infected animal, then you have to reinfect them to the second round. If the particular organism they're producing the similar kind of disease, then then they can conclude that this particular organism causing a disease. Is a Koch postulate theory. But in this case, you cannot Koch postulate. You cannot do that because this virus cannot not grow. Only if you put them on the chimpanzee, they can be causing only the liver disease. But they don't know what if this is the one they causing the disease. And another problem is, this virus also have a huge amount of heterogeneity in the genome as well. Even sequencing will get a lot of, I will show you later a few slides. So that's very difficult for them to study them, because it's very essential if you know the genome. But if you want to develop vaccines or therapeutic, you want to have a proper animal models, and also you need to have a in vitro system to culture the virus. Okay, so that's what we are doing now. Coronaviruses, if you want to develop a diagnostics or if you're testing, you need a live virus. We don't have it, therefore we are using a pseudo-typed viruses to do it. Those days, there is no pseudo-typed system or no other system like what we have it at this moment. So at the time, Professor Rice started doing a reverse genetics. Actually, what he did is he cloned the complete genome, and with the 5' and he put the T7 promoter, and he made a transcript of this particular genome, then inject them on them in the chimpanzee. So what happened, unfortunately, even if we challenge them or inject them, the chimp doesn't happen any disease. Actually, they don't replicate. Nothing happened there. It was a huge hurdle, and they face a lot of difficulties find the problem. Later, they identified the real cause for that. One of the causes for this the problem is this. They have one is huge amount of mutations. We know that. Second thing is, actually the mutation is happening naturally, okay, but it's because of lack of polymer, the proofreading activity of the polymerase, they make a lot of mutations. It's a natural mutations, they huge amount of mutations. But he identified several mutations. He corrected all these mutations, and also he found out that the non-translated region in the 5' end also essential for the replication of the virus. So for that he made several. I think here he shown here 10 different constructs with the different modifications. After that, they could modify one. When you inject them on the chimpanzee, they could able to replicate. They also have caused the similar kind of disease what happening in the humans. Actually, they have DNA. They can find out the RNA. Then also have elevated level of ALT. And also they also find out antibodies. That means they could able to seroconvert. They could also find antibodies against this this particular virus. That means if there is an elevated level of ALT, and that means indeed there was a replication. You could able to prove that. If you really interested, you can read his paper. Actually, this is old population science. Yeah. Okay. With this, actually, this was the first major achievement, because he had an infectious clone which can replicate in vitro or in vivo. If you put them on the chimp, they can able to replicate. It's a very fantastic study. Actually, really. It was a really great groundbreaking, because I don't know if somebody's work on hepatitis C virus, that if you look at the hepatitis genetic diversity, enormous. You see a huge amount of diversity. I will show some picture later now. So with the help, after that, the people able to develop, they develop, animal models, because at that moment they don't have an animal model. Now they took it, they took a human hepatocytes, they can, implant on the mice liver, then they make a chimeric mice. This mice becomes susceptible for the hepatitis C virus infection. Using this, they could be able to test that all the therapeutics and also vaccines also. And also, there are, after the discovery of the complete genome, there are the people they are able to develop different types of antiviral agent targeting different, the locations, the non-structural proteins. There are several. These are the names, but I'm not going to explain all this. But one of the problem here, I told you, is it has a huge heterogeneity, and also have different serotypes, subtypes, etc. It's very difficult to treat them. Therefore, now the WHO guideline is recommended is a pan-genotypic, genotypic direct-acting antivirals. That means you need to do, if you give a one therapeutic, it doesn't work. If you have if you really want to treat them, they want to use combination of different drugs that can target NS5A, NS5V, NS5-3, like that. The combinations will work better for this virus. And also now with the reverse genetic system, they could able to now people could generate a lot of clones. Whichever strain we have it, we can make a clone. You can also develop in vitro system. This is the virus infected cells, because people are treating antivirals agent. You see them to treat an animals completely inhibited actually. So this is the kind of system they could able to develop after the discovery of the reverse genetics. So one of the major bottlenecks in the field of hepatitis C virus drug resistance. That means that mediated by the causes species generated during the replications. Actually, these are natural resistance. I also worked a little bit on hepatitis C virus because we want to trace back what are the different types of quasi-species present in liver versus plasma. Actually, what we believe, people are believing, the reason one of the things I told you, when you give a treatment, what will happen? So the interferon, one of the example I showed you, when you give an interferon, the virus RNA goes down. If you stop giving an interferon, then appear back, So you're coming back to again in a few weeks later. So if you give it again, if you interferon, then the viral RNA goes down. If you stop it, it will coming back. Actually, this is a problem in the hepatitis C virus. So actually, what we believe that. So actually, actually somewhere else the virus has to hide, somewhere else. Actually, we believe that there is the resistance strain. Actually, is a strain inside the hepatocytes, and they once they if you give a treatment, they will be hide inside the hepatocells. Then later is coming up. Once you stop giving a treatment, they can escape from the host immunity or or particular drug. They coming back again. I also did it. I was comparing one of the drug that is targeting the NS5 is a polymerase is GDD motif targeting drug. Where we want to check back whether any naturally occurring resistant mutants hide inside the liver cells or not. So we collected the plasma samples and solo liver biopsy, and we did the deep sequencing, and we found it. The compartment device is both the liver versus the plasma was same, but we could not actually still. But however, we don't know exactly, because the liver biopsy was, we took a needle biopsy. Just to put a needle, we just suck it out, take out the small piece. But we know if you take, maybe the compartment-wise difference may be there, but we are not sure of it. So we believe that still is hiding in the virus. And also, we also proved that the virus also hide inside the exosomes as well. So actually, there is why we detected viral RNA and also viral protein inside the exosome. So that means the virus also hide inside the in the exosome, and they can also transmit it to one person to within the person. They can escape actually from the drug. Then you can transmit very easily one cell to other cell types. So if you look at the risk factor associated with the transmission of hepatitis C, actually we I think in India we don't have much, but in abroad, Europe and America, you have a huge illegal drug users there. So the people use the same needles. They also do. That's one of the major things with the people. Many of, even including jail, people use drugs, then they get infected with the hepatitis E virus or blood product. If you have a blood product, some transfusion or surgery, for example, unsafe injections, for example, And yeah, sometimes people also get mother to child contact. So of course there are many other sexual transmission also there. Now, one of the risk, and of course I told you drug is the more prevalent in the people who use drug users, not only in the syringe or needles. And this also because this is actually why it is this infection is much higher. This virus very quickly, if you use one syringe and use in another person, they can within quickly they will get it, the infections, which is which is more common than the HIV infections also. So, but when you look at the clinical feature about incubation period of initially 6 to 7 weeks, that may vary between 22 to 20 weeks as well. In the acute illness, you see a jaundice actually, so mild. Sometimes you also is symptomatic. So if the case fatality rate is very, very less, but the chronic infection is around 60 to 80%. Those become chronic hepatitis, and later they become cirrhosis, and then the people will undergo death. So what actually happened in the acute cases, actually I told you, acute cases there is a viral infections. Actually this is here you see the viral RNA titer. You see the viral RNA. Here is when you have a infection in the early months, this is a months, okay. So then you have a early months, then you see how ALT will be elevated. So meantime your immune system is activated, and they will be able to control the they're able to control the virus. But in that case, acute cases, your immune system already tackled it, and the later ALT level, because your liver become normal, the ALT level will be coming normal. RNA also will be disappear. Antibody against this particular virus will be there, so they could able to control it. But in the case of chronic cases, but it doesn't happen like this. The RNA level is still, it will be constant. The LRT level goes up, then come down, and then keep on, is a varies. So then, but still RNA level also high, even with the presence of anti-hepatitis C antibodies. So why this is? Because I told you, this is a compartmentalized differentiation. Actually, hepatitis C virus make a lot of quasi-species like this. You see, this is a naturally occurring. Okay, the mutations, there is no selection actually in the early. During the replications, the virus get mutated. You have a randomly mutated, you have a lot of quasi-species there. So one of them, for example, one particular mutant among all this, that is specifically, they mutated either the receptor binding domain, or if you think about the drug targets, whatever it is, okay. So actually, if this is a naturally occurring mutant, once you have this particular mutant, then when you give it treatments, what happened? All the others will die. The one we have now had a mutant, this will escape. If they escape, then they will come, because our immune system is never recognize this particular mutant. No. So for example, epitope mutant, and or if you have a therapeutic escape, then they will, even if you give the therapeutic, they escape and they can establish their life there. Okay, that's a problem in the in the hepatitis C virus, even developing therapeutics also. Okay, so if you look at the history of the hepatitis C virus infection, for example, if you think about 100 people have hepatitis, around 85% of them undergo chronic infection. Among this 80%, around 20% go for a cirrhosis. Around within the 20%, around 25 go for a cirrhosis. Later stage is is the end stage hepatitis will undergo death. Okay, but this I told you earlier. This process is not like if you have a cirrhosis, it will take. Actually, if you have a chronic cases to become cirrhosis and the end level cancer, then you will be cancer. Then you die. It will take around death. Actually, from chronic to death, it will take to 25 to 30 years. Quite a long incubation period. Okay, so but if somebody is having any reach to cirrhosis or or cancer, the stage you can do a therapeutic. Therapeutic is no therapeutic. Only way you can do it, you can use for the liver transplantation. However, whoever give a treatment, then liver transplantation, that they will reappear again a few months later. I think in the case of hepatitis C, there is a success rate is very, very less. So chronic hepatitis C case promoting, actually it becomes severe. So mainly in the alcohol intake, or sometimes you have a very severe in the HIV co-infections, or if somebody have a chronic hepatitis C infection, then will become more severe. And vaccines, there is currently no vaccines available. Actually, the vaccines doesn't protect, because the virus is still, due to the high heterogeneity. You see, for example, here I put the one example. So initially you have a genetic diversity of the virus like this. When you have the source initial, for example, if you have a source, when you inject them in a mice, or if you inject them in a monkey, you could estimate the diversity like this. When in the time goes, you see the progress, the virus change within the case. For example, now what is happening in the coronavirus outbreak is happening in, virus replicate in one person, the virus jump to one person to the another person, then the second person they move to the third person. Okay, then you see a huge diversity. Okay, it's a host shift, and you see a lot of changes there. But in the case of hepatitis C, not like that. It's within the same host. If you having initially you find a virus like this, the end of the day, if you find it, it's something else. I did myself. I experienced this. I could see that differences. Okay, so this is that's something very difficult to. If you have such as the virus, very difficult to make a treatment. Okay, so that is the reason. The three people got the Nobel Prize. What they did after the discovery of hepatitis C virus, what they do, they could able to screen hepatitis C viruses, and also they identify drug users, and also they could able to develop drugs. So finally, they could able to they bring back to the is actually huge decline after this discovery of hepatitis C viruses. Actually, not hepatitis C virus. Okay, you see, this is a report of 2000, but still actually the same. That's the reason I put this one actually. Okay, so actually to summarize this, actually why they got it a Nobel Prize, because is they could, since they they discovered the virus, because of that they could able to save the life of many millions people. And also this the author person, he's the one say this is something is a non-hepatitis A or non-hepatitis B is causing this type of hepatitis. And Michael Houghton discovered he's the one discovered the hepatitis C virus, and CIRRHOSIS discovered the reverse geneticism. The combination of all of together with all their work, they could able to control at least they could able to control the virus. Think about it. If you don't have any vaccines or any proper therapeutics, so very difficult to control. No, if they wouldn't identify now, is they think about how many people might have been infected with this virus. That is the reason they got 3 people got a Nobel Prize for their the excellent discovery. So thank you for the I'm going to stop now. So to thank you for the attention.
I would like to thank Dr. Stalin for the great talk. It was really informative. Thank you, sir. I would also like to thank you all for attending the session, and I hope you all will be back tomorrow for the next talk. Thank you.